I think we'll get started with the introduction, and people will still join for the next few minutes. It is a great pleasure to introduce Laura Huckins, who's been with us for a few years now. She's now currently an associate professor in genetics and in psychiatry. She does a lot of critical work on eating disorders and PTSD. She's actually the lead for the PGC Eating Disorders group. And does a lot of work also with electronic health records and GWAS from EHR, and has done a lot of very interesting work on annotating variation using postmortem samples and things like that, and also incorporating orthogonal data sets into GWAS to kind of help understand functionally what we see in genome-wide association studies. I just have a request here from Miguel about testing the microphone. Miguel, if you haven't heard me, now would be the time to say so. But if we're okay, let's just go ahead. So Laura, I'm looking forward to your talk, and thank you for agreeing to do this. And yeah, I will turn it over to you.
Thank you so much. Thank you for having me, and perfect introduction, because I think my talk is going to cover pretty much all of the things that you just touched on for my lab, so that's great. So for the next hour or so, I wanted to talk really about eating disorder genetics, kind of give you all an update, some of the things that my lab has been working on and that the broader field has been working on, and then also touch on our agenda for the future. And this really is the agenda of kind of the PGC ED working group, what we think is important and how we're going to try and redesign our studies so that we can increase diversity and increase some of the insights that we get into eating disorder genetics. So to start off, I wanted to give you a little history of eating disorder genetics over the past decade or so. We had our first GWAS in 2014. As you can see, it was not an exciting GWAS. It was not like the first schizophrenia GWAS that gave us those 108 loci. We really had a Cambridge plot rather than a Manhattan plot, not many spires. And this is with about 1,000 cases or so. The exciting thing about this GWAS, though, even though we had no genome-wide significant hits, is that we had this interesting mix of psychiatric and metabolic pathways popping out. And this really reflected what we'd been thinking about a lot in the field at the time, which was, should we be classifying anorexia as a psychiatric disorder or a metabolic disorder? And we've long had hypotheses about the fact that it must be both, right? Most people in America will go on a diet at some point. Most people will not be successful in that diet. And a large part of that will be due to psychiatric and metabolic reasons. We feel hungry. We don't like dieting. It's unpleasant to feel hungry. However, this doesn't seem to be happening in people with anorexia. They are able to lose weight quickly, and they don't report any of the negative emotions that accompany it in people who aren't able to diet as easily. So there's clearly both metabolic and psychiatric factors, and our early GWAS supported this as well. 2017, we repeated our GWAS, this time with 2,000 people, and still found nothing genome-wide significant. In about 2019, we had our first genome-wide significant locus here on chromosome 12 for anorexia nervosa. This included both genes expressed in the gut and genes expressed in the brain. So we're pretty excited to still have this metabolic and psychiatric underpinning here. And then finally, let me just move my Zoom window so I can actually see my own slides. Finally, a couple of years ago in 2019, we had the first really kind of blockbuster GWAS for anorexia nervosa, where we identified 8 risk loci. And this includes just over 12,000 cases and tens of thousands of controls, including from a number of large biobanks, such as UK Biobank. And again, we're really seeing this metabo-psychiatric origin for anorexia nervosa. This was really exciting for my lab because, as you heard in the introduction, we are interested in annotating GWAS with functional elements to try and understand the biology underlying these disorders. So we looked at this GWAS and thought about what are the big outstanding questions in anorexia and what should we do to go from these genome-wide significant hits to biology and to function. And I like to illustrate that using this kind of schema. So I think the first big question underlying this GWAS is how do we translate these SNPs through to genes and then later through to publicly available kind of candidate genes, tissues, and pathways, right? How do we go from GWAS to biology? In our lab, we do this by annotating our GWAS hits using eQTLs, and you'll see this in the next slide. The other big question that we have outstanding in, I think, anorexia, but more broadly in psychiatric genetics, is how do these SNPs affect our patients? What does it mean in a clinical population to have one of these variants or the other? How can we tease apart who might have a better or a worse course of disease, or who might be suitable for a specific treatment based on SNPs and our GWAS findings? And I'll talk later about how we can look up our anorexia GWAS SNPs in the Mount Sinai Bmi Biobank to try and tease apart what the different facets of the disorder might be there as well. The other big question that we have with anorexia nervosa specifically is BMI. And you'll see that all the stories that I'm about to tell you about work that we've done with anorexia has BMI as kind of an underlying element. I told you already that people with anorexia have to be good at losing weight in order to develop the disorder. What we don't know is whether that is a cause of the disorder or if it's a symptom. Is low BMI somehow causative? Is it having an effect on gene expression, or is it simply resulting from the psychiatric factors that underlie the disorder? So we'll pick that apart a little bit as well, using both our GWAS and our EHR later on. Okay, so to start off, a huge shout out to Jessica Johnson, who I think is one of our participants today and who really led this first analysis. So the first step, as I said, was to go from our GWAS to genes and tissues. To do this, we apply a method known as Predixscan. Others might know it as TWAS. Essentially, the idea underlying this approach is that a large portion of our transcriptome, of our gene expression, is genetically regulated. We refer to this as genetically regulated gene expression, or GREX, and we can predict genetically regulated gene expression from genotype. We're not thinking about any portion of the transcriptome that's environmentally mediated or that might be affected by other factors such as batch effects or RIN or anything like that. We're just focusing on that portion that is genetically regulated for now. The idea is that we take a large set of genotype data for which we have access to both genotype and transcriptome, and we can build predictor models linking the two data sets. So in this instance, I have three very straightforward individuals. They only have 3 SNPs in their genome and 1 gene in their transcriptome, and we can use them to build this predictor model. We have a certain weight of the dosage of SNP1, SNP2, and SNP3 that combine to predict expression of the gene here. The utility of this is that when we have access to genotype data for which we can't access the transcriptome, let's say these are living individuals and we want to be able to estimate gene expression in all of their tissues simultaneously, we can't, of course, go ahead and sample those tissues. Instead, we can use those predictor models that we just built to calculate imputed gene expression. We might do that here for these 3 individuals again. If we also know something about these people, let's say individual 2 is a case for some phenotype that we're interested in, then we can perhaps calculate from these imputed transcriptome results that gene 2 is associated with being a case. We immediately have more biological insight here than we do from the SNPs alone. Instead of saying a specific SNP is associated with disease, we can say imputed gene 2 is upregulated among whatever tissue type it is that we're interested in of cases compared to controls. So we get a gene, we get direction, and we get a tissue. We did exactly this with that anorexia GWAS that I showed you before with our 8 genome-wide significant hits. So for all 17,000 cases, we calculated genetically regulated gene expression using those models. We did this for the dorsolateral prefrontal cortex and 11 brain regions from the GTEx dataset. We also did this for subcutaneous adipose, for endocrine tissues, and 12 digestive and gastrointestinal tissues. So we're targeting specifically brain and metabolic tissues here because of those earlier hypotheses. And we then calculate our case control associations with gene expression, exactly as we did in that toy example. And this is the Manhattan plot that we get out at the end of it. We have immediately, as I said, more biological interpretations here. So instead of our top SNP on chromosome 3 being an alternate allele that has a higher frequency in cases, we can pinpoint a gene and a tissue and a direction. And they look like this. So we're able to say things like WDR6 is significantly upregulated in the skin of individuals with anorexia, or FBLAM1 is significantly downregulated in the liver of individuals with anorexia. So we get genes, tissues, and directions instead of just SNPs. These are the first genes to be identified to be genome-wide significant with anorexia, and you can see that we have a nice mix of tissues that are associated with adiposity, with metabolism, and with the brain as well here on the right. This is really interesting. We're really excited to find genes that are associated with anorexia, but our GWAS are inherently limited, and the amount of information that we can derive from a GWAS-based study is always going to be limited by our GWAS design. So as you all know, our GWAS consist of case and control populations. We try and maximize power by maximizing the numbers in each of those groups, but it doesn't really represent what happens in the clinic, right? In the clinic, we're much more likely to see this widespread of phenotype definitions and expressions. And you can imagine here that the bright pink individuals are our anorexia cases, the bright blue are our true controls, but we're also going to have a lot of other people in the clinic who have subthreshold presentation of anorexia here in light pink. Perhaps they have atypical AN, perhaps they have bulimia nervosa, they share some of the symptoms, but not all. We might have individuals with related but different disorders. Let's imagine these are other psychiatric disorders that share some genetic underpinnings with anorexia. And then we'll also have some individuals here in light blue who we maybe don't consider to be full cases, full controls, sorry. So they aren't completely healthy. Let's say they've had some experience of dieting, maybe some history of weight-related bullying. Sorry, my slides are advancing without me. You know, they have some reason that we don't consider them a perfect control in our GWAS. What we would like to do is understand whether any of the genetic variants that we identify in our GWAS are associated with the phenotypes that we see in our biobank. So if we were to order everyone, let's say by their expression of a specific gene from our study, does that reflect an ordering from extreme presentation all the way through to case, all the way through to control status, or are they generally kind of not related? Do we have any ability to use the genes that we've just found to differentiate outcomes in our clinic? So we use data from the Mount Sinai Bmi biobank to answer this question. It includes around 30,000 individuals. Bmi is unique in that it's a highly diverse data set, including, as you can see here, roughly 1/3 each of African, European, and Hispanic American ancestry. They are relatively overweight overall, so the kind of BMIs are on the higher side. This may limit our ability to capture some of those anorexia-specific phenotypes that we might be interested in, but these are the data we have, so we make do with it, but just kind of bear this higher BMI in mind as we go through these results. So we took every gene that we found associated with anorexia in our original Predixscan study, predicted the genetically regulated gene expression in Bmi, and tested whether that was associated with any phenotypes. And this is called a phenome-wide association plot. Basically mimics a Manhattan plot. On the y-axis, you still have your degree of association. On the x-axis, we have groups of ICD codes. So these are grouped by their type of disorder. And you can see that we have a number of significant associations between our genes and phenotypes. Interestingly, our strongest association is driven by a gene called CLIC1 with type 1 diabetes. The same gene that drives an association with celiac disease. This is particularly interesting because there is a substantial proportion of anorexia cases who will complain of gluten intolerance, of discomfort when eating. We think this might be partially because they truly have underlying celiac disease. We also see genes that predispose individuals in our clinic to other psychiatric traits. For example, SLC26A6 here, which is associated with MDD with psychosis. A couple of genes with sleep apnea, and then a number of genes that also predispose to what we would think of as gastric distress or gastric discomfort conditions. So we've got associations with peptic ulcer, and another one that should be coming up there for diarrhea and gastric distension as well. So there's a pattern of our anorexia-associated genes predisposing to psychiatric phenotypes and to gastrointestinal distress and issues with diet in our biobank. We also see associations between our anorexia nervosa genes and weight of our patients. So this is CLIC1 here, that gene that was associated with type 1 diabetes. You can see that as CLIC1 genetically regulated gene expression in spleen increases, The patient's weight also increases, and their risk of anorexia in our previous study decreases, exactly as we would expect. So more expression of this gene, less likelihood of anorexia, more likelihood of a higher weight in kilograms. So all exactly as we would expect. The next thing we wanted to know is whether the associations between the genes and the phenotypes that our patients present with might be altered by the patient's BMI. So I mentioned before that our patients on average have higher BMIs. The CDC would consider everyone slightly overweight, I think, by the Bmi BMI measures. We wanted to know if that would affect any of the phenotypes that we see. So we partitioned our cohort into low, medium, and high BMI, as you can see here. And we did this using a data-driven approach. So we're not placing any kind of CDC guidelines on this. We're just using the bottom quartile and the top quartile to define low and high. And you can see the numbers down here. The high BMI is really very high. The low BMI is just under 22. For reference, we usually use 19 or so as an expected anorexia cutoff. So this is a little bit higher than the clinical definition for anorexia. We basically repeated our PheWAS to see if there were any differences in these three groups. I don't have time to go through all of the results here, but I think the most interesting one is what we see with regards to chronic pain. So when we look in the full cohort, we see only one association between our gene expression and knee pain here, not particularly exciting. When we look specifically in our low BMI group, however, we see these peaks shooting up where we have associations with pain in the feet and generalized pain, as well as ankle pain. Some probing through our ICD records implies that this is probably due to foot and ankle injuries. We think this is likely due to excessive exercise, and that these genes that are associated here in the low BMI group may be predisposing either to osteoporosis, which we know is an endophenotype in anorexia, and thus these individuals are more likely to suffer foot and ankle injury when they do exercise, or it could be that these genes are predisposing to an increased level of exercise itself, and hence foot and ankle pain and injury. So this is one of many examples. Some other exciting examples included differences in substance use rates, differences in tobacco use, alcohol use, all those kind of things with BMI differences as well. But this, I think, is the coolest, most unexpected result that we found. You can read all about this study in much more detail. I didn't have time to go into sex differences in our PheWAS, but we also looked at partitioning our biobank by sex and then looking at how those PheWAS changed as well. It's just come out in Psychological Medicine a couple of weeks ago and led by Jess Johnson in our lab here. Okay, I started to touch a little bit on how BMI can change the associations in the clinic and how the BMI of our participants might alter what kind of phenotypes they express. We wanted to go a little bit further in the lab and think about whether the BMI could actually be changing directly the gene expression of the individuals in our study. I told you that we're predicting the gene expression from genotype. That makes a lot of sense if we have a static individual, a post-mortem brain. But really, as we go throughout our lives, as we experience our environments, as we interact with our environments and various stimuli, our gene expression changes. Our gene expression is not purely regulated by our genome. You saw in my very beginning figure that we have an environmentally mediated portion of our transcriptome as well. So now as a lab, we want to go further and think about what if we model both the genetic and the environmental regulation of our transcriptome? What if we do that simultaneously? Can we find any genes for which not only is the genome regulating the gene expression, but what if the person's BMI is regulating the gene expression as well? Can we find instances where we have BMI specific or BMI dynamic EQTLs, where the EQTL changes its status or changes its effect on the gene as the individual's BMI changes? So all the work I'm going to show you in the next few slides is kind of brand new, hot off the press, unpublished work from Rebecca Seiner in my group. She's a PhD candidate, brand new PhD candidate, and she's going to achieve really great things over the next few years. So we're excited to see where her PhD takes her. And this is just kind of a little preview snapshot of what she's been working on. So as I said, her goal is to understand the role of BMI in cis EQTL function, and she put together this nice diagram that I think summarizes this. So a standard EQTL looks like this on the left here, right? You've got this relationship between dosage of your alternate allele and gene expression. So here you've got this nice linear relationship. If you have an interaction model, this linear relationship will be governed by BMI. So you'll see different relationships between the alternate allele and gene expression at different BMIs. And you can see here, there's no relationship between gene expression and BMI for the AA, but there's a positive relationship between gene expression and BMI for the AC allele. So here we have an interaction QTL. It's also possible that we'll have categorical changes. So when we think about how BMI is genetically regulated, there is evidence that BMI is continuously regulated. So the more genetic risk for high BMI you have, the higher your BMI is. But there's also studies showing that the edges of the BMI spectrum are regulated differently. So having extremely low BMI or extremely high BMI may have different underlying genetic facets. So as well as modeling kind of a continuous interaction between BMI and gene expression, Rebecca is looking at what happens at those extreme ends, at those people with incredibly low BMIs. This is in the GTEx cohort. And you can see there's some people with a BMI as low as 15. We would hospitalize someone for this BMI, right? This is extremely unusual and low. And we can assume the genetics here are different than the genetics in the middle. So she's also partitioning the cohort to the lowest and highest and looking at what the EQTLs look like there. So we'll have BMI dynamic and BMI specific EQTLs. We find a surprisingly large number of EQTLs with evidence for BMI interaction. You can see here, this is most of the genes that we test in most of the tissues have at least one significant interaction QTL. The leading tissues are all brain, which was really exciting and interesting for us. Those of you who played with the GTEx data set in detail will know that the brain tissue are not the largest. We would usually expect that the largest tissue has the largest number of E genes. That's not the case here. This is really driven by brain-specific effects, as well as metabolic and adipose tissues, as we would expect. And somewhat surprisingly, testis here in fourth, and I don't have a good explanation, except to say that testis in general has more E genes than you would expect for its sample size. But we have no idea why there's a BMI effect going on there. And you can see here that there's no relationship overall between the sample size of the tissue and the number of BMI significant E genes. So we think there is something truly biological going on in which tissue has the most E genes. It's not just a power thing. We have a really nice distribution throughout the brain of where our BMI dynamic E genes fall. So this is just kind of showing the majority are in neuron-rich regions of the brain, as we might expect. But we also have a nice number of BMI dynamic genes kind of in all of these psychiatric relevant tissues, which was really exciting to us as well, especially the amygdala, which is one of the kind of brain regions that we've been wanting to investigate for anorexia for a long time. If we look at which genes within our anorexia association have this BMI dynamic effect going on, you can see there's a very large number of our genome-wide significant genes have at least one BMI QTL. If you go all the way down to nominal, we also have a significant enrichment of BMI QTLs compared to what we would expect by chance. Why are we looking all the way down at nominal? This association is all based on the static BMI, the static EQTL inference of gene expression that I talked about at the beginning. Once we take BMI into account, we hypothesize that these associations might change, right? So this is all based only on the genome. If we also take into account low BMI, high BMI, and recalculate these gene expression levels, we hypothesize that some of these genes here at the bottom may shoot up in significance. So that's kind of the next thing on our to-do list to see how this would change if our participants were to be of low weight, high weight, medium weight, and so on. I wanted to pull out a few of these exciting examples at the top just to kind of show you what that looks like and why I think this is interesting. This is one of our top hits here, which is REH2 in the colon. you can see that this is a BMI specific EQTL. So this EQTL effect really only exists in individuals of extremely low weight. So presumably dysregulation of this gene occurs in individuals with anorexia, perhaps later stage in the disorder. There's not really much dysregulation going on in individuals of normal weight. We see a similar effect in this NDU-FAF3 gene, where again, we have an EQTL that's more or less specific to low BMI, although interestingly, there's a small and opposite EQTL effect in individuals with high BMI. So this implies if someone has the specific SNP here, if they've got the T allele, and they are of low BMI, they'll have upregulated gene expression. If they're of high BMI, they'll have downregulated gene expression. So potentially as their BMI changes, as they gain weight, their expression of this gene decreases. This is obviously going to be hyper relevant to our understanding of anorexia nervosa as a disorder, not just with regards to its development, but also with regards to recovery and what happens when individuals start to gain weight and leave this low BMI state and move towards a kind of medium or high BMI throughout their recovery journey. This effect is true across a lot of genes. This is another kind of interesting example here where we've got an effect at low BMI, but a more pronounced effect at high BMI. So again, as an individual gains weight, the expression of this gene will change. In this case, it'll become even more downregulated. And here again, we've got an opposite effect between low and high. So as this individual gains weight, their expression of the gene will increase. And the same here as well. Okay, so I wanted to pivot now from talking about EQTLs and context to talking about clinical phenotypes and electronic health records. This is kind of the other side of what my lab does. And I wanted to focus again on BMI and weight trajectory, but from a slightly different angle. So this work has been led by Dr. Jiayi Zhu, who's a postdoctoral fellow in my lab. And this really has been completely kind of derived and led by her. So we're very excited to see how this has evolved over the past year or so. Her idea was that weight data in the EHR is this kind of huge wealth of data that is typically underutilized. It's collected at every single visit. That is pretty unique to the US. It's pretty rare to have this large number of weight measures for patients. And in Bmi, we have somewhere between kind of 8 and several hundred weight measures for each of our patients over a course of many years. So we've got this kind of untapped wealth of information about how people's weight changes. Okay. Her goal was to derive or to find out how adult weight changes throughout their lifetimes. And this is kind of a hypothesis-free approach. So just to try and model what different trajectories we have in the biobank. What we found is there are 6 different profiles of weight trajectory throughout adulthood. Most obviously, we have stable weight, weight loss, and weight gain. We define weight loss and weight gain as a change of either more than 5% or more than 10% from the starting weight. But we also see these patterns along the bottom here of weight cycle. So you can think of this as yo-yo dieting, weight loss and cycle, and weight gain and cycle. So these are kind of the 6. Each of these, I think, has several thousand individuals in Bmi falling into them. And we found that they're all genetically heritable, which was a pretty exciting finding. To test whether these are kind of phenotypically reliable, whether these follow the patterns that we would expect, we performed a phenome-wide association study, so testing what phenotypes individuals in each of these groups were enriched for. We found that individuals with a weight gain trajectory in adulthood, as you would expect, are enriched for morbid obesity phe codes, as well as things like obstructive sleep apnea, other sleep apnea, respiratory system disorders, edema, exactly what you would expect. This, to me, is very much a kind of proof of concept and positive control. We find that weight loss trajectory is associated with a decreased risk of sleep apnea, obesity, and an increased risk of things like gastrointestinal complications. Again, exactly as we would expect and more or less a positive control. Interestingly, we also see some associations with substance use, dementia, and neurological disorders as well with weight loss in adulthood. Weight cycle, I think, was less of a positive control kind of analysis, and here we genuinely didn't know what we were going to find. Interestingly, we saw the weight cycle was uniquely associated with major depression and mood disorders, and a bunch of other psychiatric conditions. So this kind of weight cyclic behavior was really pretty much the only type of weight trajectory where we see an enrichment for mood disorders. And this is not caused by any medication use. We corrected for all of the antidepressants that we had information for. And we have pretty extensive medication information for people in Bmi that doesn't seem to be causing it. It seems to be something else. Perhaps it's the mood of the patient themselves, and episodic presentations of their disorders are actually causing these weight cycles. The only associations that we see between psychiatric disorders and weight loss, as you can see, are substance addiction and tobacco use. I should note we don't have enough individuals with anorexia in Bmi to expect to see a significant odds ratio. Because we might expect eating disorders to be significantly comorbid with all of these, we removed everyone who had even one diagnosis of an eating disorder, and these odds ratios remain. We also replicated this in the UK Biobank and see the same patterns, the same types of trajectories, and the same associations with all of these Phi codes as well. Okay, our next question, because of course our key interest here is anorexia nervosa and anorexia genetics, is whether anorexia nervosa PRS significantly predisposes to adult weight loss, even among individuals who are otherwise psychiatrically healthy. So does having a higher polygenic risk score for anorexia make it easier for you to lose weight, even if there's nothing else about you that has anything to do with anorexia? And if indeed our earlier hypothesis about people being predisposed to anorexia because dieting is easy, if that's true, then we should see a positive association here. And we included as a positive control an obesity PRS from a relatively recent BMI GWAS. A lot of BMI GWAS include Bmi, so that's why we're using a slightly older one that didn't have Bmi cases in there as well. So our positive control first was successful. Obesity PRS is significantly associated with weight gain trajectory, as we would expect. And interestingly, anorexia nervosa PRS was associated with weight loss trajectory. So indeed, it's true that adults who have no anorexia, no psychiatric codes, but with higher anorexia PRS are more likely to have weight loss in adulthood. This was actually true across Europeans and our African American cohort. It's pretty unusual to see a significant PRS association with African Americans, but I guess the result is kind of strong enough that we see it reflected here as well. And we see the same direction of effect in our Hispanic Latino cohort. It's not significant, but we think that's probably because the base cohort is completely European, not because the effect is not true in this group as well. As I said, we replicated this in the UK Biobank, and the association with anorexia PRS also replicated very nicely in the UK Biobank. We compared the top and bottom decile there rather than doing the dose response because we had less data. But again, we see that higher AN PRS is associated with weight loss in adulthood in UK Biobank too. That's actually important because those individuals aren't patients necessarily in a healthcare system. So it's not showing us that higher AN PRS just means you're sicker in general and that's why you're losing weight. It's an independent association regardless of kind of disease or anything else like that. This work is all on bioRxiv. It's actually now in press at Lancet Digital Health, so that'll be out in the next couple of weeks as well. So feel free to have a read of that and get in touch if you would like to collaborate on this. I know GIE is always really interested in thinking about ways to expand this analysis and think more about weight in EHR. Okay, for the final portion of my talk, I wanted to pivot a little bit and think about what next for eating disorder genetics. I think it's kind of been clear through this talk, there's a lot of interesting different angles for the eating disorder field. We're thinking a lot about the role of weight and environment and different factors in eating disorder genetics. And we kind of synthesized this all in a review that actually came out a couple of days ago in Molecular Psychiatry, so you can kind of read about where the field will be moving next there. And this is also partially the work again of Jessica Johnson and Rebecca Seiner from my lab. So thank you to them for all their work. So here's the citation for the review if anyone's interested in reading it. So we took as the underlying principle of our review what is known as the Academy for Eating Disorders Truth Number 5. So the Academy for Eating Disorders, or AED, put out this long document of truths about eating disorders that we should be discussing and kind of accepting as a field. And this one really spoke to us and seemed like an important thing that we should reflect and write a review about. And it's that eating disorders affect everyone. Basically, they can affect people of all genders, ages, races, ethnicities, body shapes and weights, sexual orientations, and socioeconomic statuses. There are a lot of myths and stereotypes and misconceptions surrounding this that I think have permeated the way that we think about eating disorders, the way that we diagnose eating disorders, the way we portray people with eating disorders in the media, and they consequently has had a harmful effect on people's diagnostic journeys, their ability to get treatment, and their inclusion in research. So we have this kind of snowball effect of excluding people from diagnosis, excluding people from treatment, and hindering our own ability to learn more about the disorder, which consequently makes the stereotypes worse. So this review is kind of an attempt to address that and to move ourselves back onto a more inclusive path and to design better studies. We started by looking at an overview of all of the eating disorder genetic studies that have been completed, I think, since 1974. You can see that the majority of studies have focused on anorexia nervosa specifically, or have been a kind of cross-disorder approach. Most studies have looked specifically at women only. It looks here like a lot of studies include male and female participants. When you go into the data in more detail, you'll see that those are primarily candidate gene, and that the GWAS and TWAS studies have relied mainly on female-only data. We are trying hard to change this, but the truth is a lot of the kind of existing GWAS data only includes female participants, so it's very difficult to kind of fix the existing data, basically. Almost no studies have focused on men only, and those that have, again, are almost exclusively candidate gene studies. And the same story is true when it comes to race and ethnicity. The majority of our studies have focused on Europeans, as is true for most psychiatric disorders. A small number of studies, including many of the early GWAS, do include non-Europeans. There's been a large cohort of our colleagues from Japan and Taiwan have collected large samples. And there's also a very impressive effort from the LAGC consortium to increase sample numbers, but they're not included in our GWAS yet. So this portion we expect to change in the next couple of years. This portion is going to be slower to improve, but I just wanted to kind of highlight how bad the inequities in genetics research are. These are kind of my three favorite sentences from the review, and if you take nothing else home from this talk, this is really what I think is important to know and think about with eating disorders, but perhaps with psychiatric disorders more generally, which is that our failure to acknowledge diversity has led us to overly narrow definitions of disease and diagnostic criteria. And those failures are compounding, right? So individuals who don't meet our stereotyped criteria are excluded from research and treatment. Our stereotypes are propagated without malice, without intent, but with very serious and damaging consequences by the media and by our clinicians, which contributes to underdetection and undertreatment of individuals who don't match our stereotyped preconceptions. So how do we fix this? What do we do as a field to try and move towards a better future and better studies? The first thing, obviously, is broader and more inclusive recruitment. This will mean increasing the diversity of individuals we recruit into our studies, obviously. It will also mean thinking about different ways to recruit people into our studies, thinking about how do we recruit teenagers? How do we recruit people in midlife? How do we convince people at home that they should volunteer for a study? It will mean changing our study imagery, our study marketing, so that it includes pictures of people that look like the people we're trying to recruit. It will mean having scientists working on the studies who, again, look like the people we're trying to recruit. If we don't diversify our workforce, we can't diversify our patient recruitment. It will mean explaining our studies in different language, so in language that is understandable to our patients and is understandable to lay people. We've had a lot of success over the years with apps in the eating disorder community. We've had a lot of success with social media. Those kind of things will need to continue, as well as kind of community-based outreach and engagement. So it's moving beyond the kind of basic patient recruitment towards how do we recruit the people who aren't patients because they don't feel included. Building capacity, this will primarily focus on expanding our workforce and expanding the understanding of our clinicians and our researchers about what eating disorders are. I think there will need to be a lot of work together with funders, with the NIH, with NSF, with people like the One Mind Foundation, BBRF, to redefine the kinds of questions we should be asking about and the questions that we should be funding in eating disorder research. It's not just about understanding anorexia nervosa. All of the data that you saw me present today focuses on anorexia nervosa because that is what is primarily funded. But there's many other eating disorders, bulimia nervosa, binge eating disorder, ARFID, that are chronically underfunded by these bodies. So kind of building our capacities there, building out our workforce will be key to more diverse studies and understanding. I think it's pretty clear that I think our diagnostic guidelines are biased, and consequently our understanding of eating disorder disease nosology is kind of shaky at best. Without having appropriate diagnostic guidelines that aren't influenced by bias, we can't possibly rely on the genetic results that we get from our studies. They're always going to be curtailed by the people that are included. And this will include, about 5 years ago, we removed amenorrhea as a criteria. So the idea that someone should stop menstruating and that that's part of the diagnosis for anorexia. This is obviously biasing clinicians against people who don't menstruate, whether it's people who are already menopausal, people who are prepubescent, people who don't menstruate because of their sex or their hormones. Even though that can be a big caveat in your diagnostic guidelines, if you have the word amenorrhea in front of you, it biases you towards people who menstruate when you're making your diagnosis. This is one example of many other examples of biases kind of seeping in to anorexia and eating disorder guidelines. And I think we're ready for a really big restructuring of how we diagnose and define those disorders. In parallel to this, and this is kind of where my work comes in, is I think we need more sophisticated analytical approaches to refine phenotype-genotype associations. I talked a little bit in this talk about the role of BMI and the role of changing weight and whether weight is a symptom or a cause of anorexia. And I don't think we really have a good answer for this, but we do know that when we start to think about BMI, it changes everything we think we've found, right? We can find our GWAS associations, but they'll change when we remember that our patients' weight will change throughout their lifetimes. So what does it mean to have a static association in a dynamic human living being that interacts with their environment or interacts with their clinician? We're going to need approaches that can model that can model how the physiological environment might affect gene expression, how recovery might affect someone's gene expression, how different medications might interact with BMI, all these questions. In parallel, we need to think a lot about things like sex and how things like puberty, menopause, pregnancy, how all of those will affect hormones, affect gene expression, how that might alter onset of eating disorders and trajectories of disease. So this is going to take a lot of very careful functional genomics work and integration with GWAS to understand the impacts on disease associations as well. Finally, this kind of goes hand in hand with the previous point, that we need to expand our insights beyond genotype and towards biology and environmental interactions. as well as all of the functional genomic stuff that I just talked about. I think this is going to involve kind of closer work between researchers and clinicians, understanding the impacts in the clinic of some of our associations, and more of that kind of FWAS type work to really tie together what's happening in our patients with what's happening in our genetic studies. Okay, thank you very much. That's all of my slides, and thanks to Jessica Johnson for this wonderful illustration as well. She's our kind of lab artist. Thank you to our funders, especially the Klarman Family Foundation and NIH. To our collaborators, especially the PGC, as Joe mentioned in the beginning, I'm co-chair of the PGC Eating Disorders Working Group. All of the genetic data that we've used here today comes from studies that they've put together over the past decade. And especially thank you to my wonderful lab who make working fun. And with that, I'll take any questions.
Thank you, Laura. And I probably should have said this earlier, but you're also you lead you're the director of the genetics and the MTA, whatever it's called, genetics and data sciences, right?
I am, yes.
It comes through in your presentation because it's so clear.
Thank you.
So if you have questions, you put them in the Q&A box. Those who don't know that yet. Maybe I can start. When you show the click one versus weight, I mean, that was pretty shocking. The variance in weight, related to predicted gene expression. So what is the variance in gene expression?
Yeah, so that's a good question. Click one gene expression in Bmi. So we threw out genes that had a variance that was too low. So when you do genetically regulated gene expression, you're really looking at a combination of minor allele frequencies of all your EQTLs, right? So we threw out those genes that rely on 1 or 2 EQTLs and that give you this funny trimodal pattern. So it's relatively normally distributed, and they're all scaled to be between kind of -2 and +2. So I don't know the exact variance off the top of my head, but we squeeze out the ones that fail at the bottom. So the variance isn't too low.
Yeah. So the click one expression, many QTLs are contributing to the chain, and I mean, just as a simple-minded biologist, I'd love to know if it's a 2-fold change or 2% change, right?
Yeah, and I can't tell you. Sorry, I can definitely look it up and let you know.
But the weight change was, I mean, massive.
It's pretty staggering, and I should say the other really cool thing about click one is that it's an MHC gene. So it's our first MHC gene associated with anorexia. But given that it has a role in type 1 diabetes and also celiac, maybe that makes the change in weight less surprising, right? Because if it's having a big role in those things that really do affect your ability to absorb nutrients, maybe that explains the role in BMI more than, unfortunately, more than the role in anorexia, realistically.
But then wouldn't you see it just in a straight like PRS of BMI? So
yeah, that's a good point, and it hasn't come up, I don't think, in GWAS of BMI, although they probably are stricter with excluding things like celiac than if the true underlying cause is celiac or type 1 diabetes, that's not excluded from the anorexia GWAS, but one would hope or assume it's excluded from the BMI GWAS. So that could be it. Yeah.
So additional questions. I'm trying to open the Q&A. So I'm sure people have questions about this. Fascinating to know. Just got to type them. We got to take that extra effort working to type them. If somebody wants to ask one verbally, we can always unmute people.
Can people put their hands up and ask questions, or does it have to be typed?
If somebody types and says, I'd like to ask verbally, then we can be allowed to talk. We're very strict here, so we don't have any Zoom bombing. No Zoom bombing. Here's a question from Dorothy Grace. We typically think about eating disorders, and you might be able to open the Q&A too. I'm not sure, but if not, I'll read it just also for the general readership. We typically think about eating disorders, anorexia, onsetting during later childhood, adolescence, and later. But thinking about your weight data, I wonder if are any very early weight signals from very early years, like infancy, that might signal early risk for future eating disorders.
Yeah, great question. So I think I have an answer about early life and also about midlife that's relevant. So We have been thinking about signals from early life. There are two really nice papers. One is about the relationship of ARFID, so avoidant restrictive food intake disorder, and later development of anorexia. I think that all comes from the ALSPAC study. So children who are anything from picky eaters through to diagnosed ARFID are at increased risk of developing anorexia later, as we might expect. The PGCED is in the middle of putting together an ARFID GWAS that will look at that in detail for the first time. And we're hoping to ask exactly that. Is there a relationship between ARFID, PRS, and anorexia development? At the same time, some folks have looked at weight trajectories in childhood and adolescence and whether that is associated with ANPRS. So basically exactly the same study that we did in adults, they did in kids. Theirs found a lot less because I think you would be really alarmed if a child loses weight throughout childhood or loses weight throughout adolescence. So they're very much more predisposed to the weight gain or the weight cycle phenotypes than we are because, you know, most adults have stable weight. But there is a relationship between slower weight gain in childhood and ANPRS. So perhaps the same genetics underlying it, but I think they're underpowered. And I see Dorothy has asked a related question. Anything associated with infancy, such as neonatal weight issues? There are studies looking at birth weight and weight gain of mothers when pregnant. It is hard to disentangle, from my understanding, the mother's own or the pregnant person's own BMI and experience of eating disorders and their own eating disorders, PRS, from the impact on the neonate. So I think a lot of people are looking into it, but we don't have a firm answer there yet. And I would say the other side of it is actually there's a kind of growing body of work into eating disorders and anorexia in menopause and later in life. And actually also some in kind of 80s, 90s onset and people who tend to be grieving. And there's kind of a growing recognition of a lot of onset of eating disorders then as well. And it's hard to know if those were just missed when people were teenagers, perhaps because, you know, diagnosis was not as efficient 50 years ago and those people really did have an eating disorder when they were teenagers, or if it's genuinely new onset. But I think there's a lot of really exciting work coming up about that, about EDs throughout the lifespan. Okay, another question from Maria. Do you think that there is a genetic overlap between eating disorders and another type of psychiatric disorder, or is the genetic background of the former ones more distinct? Great question. So there is a significant genetic overlap between eating disorders and pretty much most other psychiatric disorders. I think there's a nice genomic SEM paper from Jordan Smoller's group that demonstrates that, the degree of overlap. Generally, people think of eating disorders as having significant shared genetic correlations with things like OCD. People have demonstrated significant genetic overlap with substance use disorders and have suggested that might be because there's kind of an addiction to food phenotype. I don't know whether I agree with that or not, but that's kind of one of the prevailing hypotheses. And there's also a lot of shared genetics with PTSD and MDD and other stress disorders as well.
Well, thank you for really a comprehensive talk and a great talk. And I really have to say to everybody that you are certainly one of the leaders in eating disorder genomics and genetic research. And as a result, I'll leave it to you to talk about what's happening next in your life if you want to.
Thank you so much. Yeah, this is kind of a bittersweet talk for me, I guess, because I'm leaving Mount Sinai in the next month to join Yale School of Medicine to start, well, kind of continue my lab there. And we'll be continuing to look at eating disorders in PTSD as well. And really hoping to kind of expand our functional genomics work and understand more about these contexts and how things like sex hormones and hunger hormones and stress hormones all influence anorexia risk. So it's going to be an exciting few years for us, but also a very sad time to leave all our friends at Mount Sinai, definitely. So thank you very much for inviting me here today. I enjoyed giving this talk.
Thank you for a great talk and best of luck at Yale.
Thank you.
We shall miss you.
Thanks, me too.